Hello and welcome back to Recovery After Stroke. Before we get started, a quick thank you to my Patreon supporters. Your support helps cover the hosting costs after more than 10 years of me doing this show solo, and it helps me keep creating episodes for stroke survivors who need hope and practical guidance. And thank you as well to everyone who comments on YouTube, leaves reviews on Spotify and Apple Podcasts, buys the book, and even to those of you who don't skip the ads. Every bit of that supports keep this podcast going. Now, today's episode is about the PESTO trial results, and I'm interviewing Professor Vincent Thijs. If you've ever felt confused by the conversation online about perispinal etanercept, some people sharing positive experiences while others feeling disappointed, and plenty of strong opinions in between, this episode is designed to bring clarity. We talk about what the PESTO trial set out to test, how the study was designed, what it found within the measured timeframes, and what the results can and can't tell us. Just a quick note, this conversation is educational and not medical advice. Always speak with a qualified health professional about your situation. All right, let's get into it. Professor Vincent Thijs, welcome to the podcast.
Thank you for having me, Bill.
I'm really looking forward to this conversation. Etanercept is one of the most hotly discussed topics in stroke recovery, and there's a lot of misconceptions about whether or not it is or is not efficacious. And while there's a lot of anecdotal evidence where some people have had positive outcomes from injections, there's also a lot of people's feedback which is very negative about their experience with etanercept injections and the lack of results. So today, the reason I reached out is because I wanted to get to the bottom of the findings of the PESTO trial, and I'm hoping that you can shed some light on that. The first question basically is, can you start by explaining in simple terms what it was that the PESTO trial set out to investigate?
All right, the PESTO trial was in response to community members, stroke survivors, wanting to find out whether the current practice of administering etanercept, as done in the US, in private practice, in Denmark, I hear there are some sites that provide this treatment, whether this treatment engenders can be actually proven according to the standards that we use in the pharmaceutical industry to get it to become accepted as a standard of care treatment. For that, you need to do what we call a randomized controlled clinical trial, preferably two, that show evidence that treatment does what it's set out to do. And that's why, with this background and the community pressuring the minister several years ago, Mr. Hunt at the time, to fund a trial that would help answer that question. A call was set out to do this trial, and several groups in Australia applied, and then an independent committee decided to award the trial to the PESTO study group. And then we tried to design this trial to give an answer. So it's mostly about people that have moderate to severe disability after their stroke, that have reduced quality of life. And we wanted to know, does their quality of life improve when etanercept is administered? And we wanted to test whether 1 or 2 injections were needed. Because that's what we heard from stroke survivors that from Australia and internationally that went over to the US. Well, this is how it's done. You get 1 or 2 injections. And there was a paper that had shown big effects with 1 injection. So that was the primary endpoint. But then we also looked at whether 2 injections could help. And when you design a trial, you have to make a decision. Will we focus on people with pain after stroke, or will we look at people who have mobility issues or speech issues or cognitive issues? And we saw that current clinical practice actually was people with various impairments after stroke were accepted and received the treatment. And what would have been the advantage of doing, say, only mobility or only pain? Well, you can then look at the outcome of pain or mobility. Does it improve? Or is your cognition improved? But because we wanted to be pragmatic, and we know that recruitment in clinical trials needs to reflect how is current practice. So we thought, let's put in all the people with moderate to severe disability, whatever their impairment after stroke, and reduce quality of life. And then we looked at quality of life as an outcome rather than an individual impairment.
Mm-hmm.
And so what we did then was to use the randomized technique, and where it's left up to the computer to decide what treatment a person will receive, the active Etanercept or a similar looking placebo, and then look at 28 days. And we had to make a decision, what makes sense, 28 days, what is practical, to see whether that injection then had improved quality of life. And then we did another injection, again with a placebo or the active drug. And then after 28 days again, we looked again whether that had made a difference. So we had people that had received 2 times the placebo, 1 time the placebo, and 1 active injection. And then we had people that had received 2 active injections. And then we were able to compare those and see whether they had made bigger improvements if you received 2 injections versus 1 or 0. Unfortunately, we couldn't show a difference in quality of life at 28 days, and we also couldn't show an improvement at 56 days after people had had 2 injections. So that was in a nutshell how we designed and the background of the study.
So the main difference then between the Griffith University study and your particular study was that they did go after a specific improvement in one area, I believe. Was it in...
Mostly, I think, yeah.
Okay. So although those guys went after pain, you guys went after just a general improvement in quality of life after the injection. And your stroke survivors could have been as far as 15 years post-stroke, is that right?
Yes, correct. We wanted to have people early after stroke, between 1 and 5 years, and then also between people 5 to 15 years after stroke. That was also for practical reasons. Once you start a trial, you see how good recruitment is, how many people want to participate in the study. And we saw that if we went to up to 5 years, recruitment was relatively slow. So we added this additional group of people later on after their stroke. So that because many people, I'm 5 years, I'm 6 years after stroke, why can't I get the treatment? And, so we also wanted to expand the pool. And that's also what happens in clinical practice. Current clinical practice, I don't think the sites in the US, and they would refuse a patient 6 years or so. We just wanted to reflect the people that we see on the website going for this treatment.
Yeah. And then the difference between the Griffith trial and your trial as well was the actual dosage of Etanercept, the amount that was in the injection. I do believe that your trial was a 25 milligram injection. And I believe that the Griffith University trial was 25 milligram injection to 50 milligram injection.
Yeah, we just based on what people told us they received when they went to the clinic, also the other sites, and then also 25 milligram was chosen because that's in the patent for this treatment.
Okay, I see. So you're trying to, as much as possible, mimic what was happening out there in the private practice.
We wanted to answer the question, is current clinical practice, is that beneficial? And that's what the call was, to do a clinical trial in current clinical practice. You have to make decisions, right? And I think this was the most relevant for stroke survivors.
Now, that's really interesting that stroke survivors were able to twist the arm of a minister to get the funding to begin that process of the trial. How long ago did this actually start?
I think it was 2016, 2017 or so. So it takes a while to get the minister, and then I think that the trial started in 2019. COVID hit, yeah, took a while to complete as well. Yeah.
Right, understood. Okay, so then you recruit people, they come along, and they go through the trial, through the particular trial. How does that work on the day? Do they turn up? Are they admitted? We'll be back with more of Professor Thijs' explanation in just a moment, but I want to pause here because if you've ever felt stuck between hope and uncertainty, you're not alone. When you're recovering from stroke, you're constantly making decisions, and some decisions feel high stakes, especially when confronting information that's conflicting. In the second half of this conversation, we get into the parts that really help you think clearly, what the trial results do and don't mean, and why placebo responses matter in blinded research, and how to frame smarter questions before you commit time, money, or energy to any path. If you want to support the podcast and keep these episodes coming, you can grab my book at recoveryafterstroke.com/book or join the Patreon at patreon.com/recoveryafterstroke. All right, back to the episode.
All right, so we recruited from a variety of sources. So we had kept a log of people that were interested in this. We had Facebook posts in New Zealand, for instance, where we recruited as well. We had people from the stroke clinical registry that were approached. We had a website and people could register their interest if they were doing a search online to participate in a clinical trial. So a variety of sources, and then we have to determine eligibility. That was mostly done either via an in-person visit or remotely via telehealth. We tried to get their medical information, whether they had, what type of stroke they had, and then we also questioned whether they had the modified Rankin scale, the disability they had, the impairments they had from their stroke, and so that then people came when they were considered eligible. Then we scheduled a visit, and they would typically come in, no overnight stay needed. It was a day procedure that was done. People were then receiving another questionnaire on the day itself to measure their quality of life, and other measures like their fatigue levels and how much help they required, etc. And then we proceeded with the injection, which was done. We had bought a special bed that was able to do the tilting that was required. So we set the people up, injected, and then tilted the table. So we received the drug. It was prepared independently by the pharmacist. So the pharmacist, they took the drug off the shelf or made a placebo, and they made sure it looked exactly alike. So then somebody from the trial team picked it up from the pharmacist. The pharmacist didn't tell, of course, what it was. And then the administration happened. So the doctor who administered and the participant did not know what they received. So after the procedure, they were left like this for 4 minutes. And then after 4 minutes, people could sit up again. And we waited about half an hour. And then we asked them how they were doing, whether there were any adverse reactions. Then after that half an hour of observation, people could go back to their habitual situation. So it's a very simple procedure to do. Yes.
I believe there was there 126 participants?
Yes, 126 people participated. We had anticipated a little bit more people to participate. So we had hoped 168, but recruitment fell flat after a while, and we were not able to find more people to recruit. So we made a decision, and then these clinical trials, they have some funding, and they require the treatment team to be paid, etc., and that ran out. So we had to stop at a certain time.
Mm-hmm. Was the study stopped early because of a decrease in the amount of funding, or was there an issue with the funding at some point?
The funding ran out. You hire people for a certain amount of years, and then you have fewer patients than you anticipate, so you have to stop.
Okay. So would that affect the outcome of the trial? Would you say the lack of funding or the lack of the ability to take the trial further?
Yeah, well, what we had when you do the trial, when you plan the trial, you say, well, this is what we're going to expect in terms of efficacy. You have to make a guess and say, well, that many people will have an improvement in quality of life if we give them the placebo, and that many people will have an improvement in quality of life with the trial drug. And we had thought that about 11% would improve with the placebo based on an earlier study. And then we had to make a guess, because nobody had done this type of study, on what Etanercept would provide. But reading the report that was published several years ago now, where 90% of the people reported improvement in their impairments, we thought, well, let's not go for 90%, but a 30% improvement. And so that was based on that. We needed 168 people to participate in the trial. So that was the what we call the pre-planned sample size estimation, which is a guess. When we stopped at 126 participants, actually, we saw that the results were very different. There was not that 11%. Actually, in the placebo arm, we saw that 58% of the people also had that improvement. Wow. And 53% had it with Etanercept. So our initial guess was very wrong based on some advanced statistical techniques. We have quite a lot of power to estimate whether there was a difference. So I think the trial can provide us an answer. It's large enough to give us an answer about this particular question. Is current clinical practice in these people with this range after their stroke, does it improve the quality of life after a month or after 2 months? I'm not speaking about early improvement. I'm not speaking about 6 months down the line. We only can decide what we see in this study.
So you have some limitations because you can't have the funding to test 1 month, 2 months, 6 months, 12 months. You have the funding to basically meet the design of your study, and then you can report on that. Now, what's really interesting is that the placebo had such a large result. What kind of things were people reporting that improved for the people who had the placebo injection?
Mm-hmm. Look, this is, of course, when we were in the blinded phase, when neither myself or my colleagues who did these scales, we were totally blinded. And that's, I remember vividly people saying, oh, it didn't do anything for me. But then there were also people said that they could see again. And some people that had improvement in sensation. Some people had improvement in their speech. So there were, we observed these things, but we didn't know whether they were active or placebo. And then surprisingly, we had some people in whom we thought, oh, they must have had active drug, that turned out to have the placebo, but that's years after, right? Because it took takes a little bit of time to accumulate a sufficient number of patients, and we were only reporting and breaking the blind when the trial was finished, because otherwise you may be biased in all your analysis, etc.
I see.
You don't want to do that. So you wait until the end of the study to break the blind. And that's very frustrating for the participants, because there were many people that said, I must have had the placebo because it didn't do anything for me. And there were other people that were, and some people like that, they said, I still want to go to the US. And please, can you tell me if I received the placebo? And I understand it was terribly frustrating for these participants. But we were very strict. No, we don't want to break the blind. This is against the rules that you have to adhere to in a clinical trial. And so we didn't do that. Of course, once the trial was finished, we were able to report the results back to the participants. And then there were some people that were very surprised that they had received the active drug. I remember one person vividly who said, you have to tell me now because I'm going. And then I said, hold off. And then we told them you had twice the active drug. And so they decided not to go anymore. So you see how from a clinical trial perspective, it's very important to remain very objective and not being able to see what people have received. How, from a humane level, of course, I understand it was very important to these people.
Yeah, that'd be difficult. And then I imagine that had the placebo not worked and then the etanercept did work, then there would have been people who would have said, well, I've received the placebo, it didn't work for me. Other people received the etanercept, it did work for them. Why can't I get the etanercept injection now?
Yeah. And we also had people that had twice the placebo who noticed an improvement and have told me the improvement is still there.
Wow.
So
that's amazing. Now, was the intention...
And often that, and I must tell you, often those were relatively little things that seem to improve both with the placebo and in the active group. And you see that there are changes in quality of life that people have reported, but it happens as well with the placebo. Yeah.
Wow. Was the intention of the study that was funded at the very beginning in 2016 by Minister Hunt, was it to determine whether or not this was going to be an effective treatment for people in stroke and therefore to roll it out somehow in the Australian medical system for stroke survivors? What was the thinking for Minister Hunt? Do you know?
Of course, I was not involved in that lobbying to the minister or anything, but it was to bring it on a pathway towards regulatory approval. We know that Etanercept is a relatively cheap drug that you can get and is approved already for some indications, especially in people with rheumatoid arthritis, the condition of the joints, but it's not approved for stroke. And to be officially approved and then potentially reimbursed on the PBS, you need to have some trials that have been done, such as PESTO. We do different trial phases. One would be a phase 2 trial and a phase 3 trial. So phase 1 is typically in people just to assess the safety and some dosages, usually in healthy people. And then a phase 2 is safety among stroke survivors and preliminary efficacy. And that's where PESTO was what we call a phase 2B trial. And then a phase 3 trial would then be a trial in many more participants based usually on the results of a phase 2B trial. And then usually when you have a phase 3 trial and it's convincing, and the authorities may approve such a trial.
So in this case, the phase 2B trial, this PESTO trial, didn't find that it's efficacious, and as a result, there's not going to be a further trial. Would that be accurate?
Well, based on the findings we have in this particular type of way of administering in this particular group of people, I don't think there is enough evidence to argue for a phase 3 trial. It may be that say you could say, well, we want to focus on pain because that was more promising. Well, you'll need to do another trial in that condition.
Yeah.
In after stroke, or maybe within a year after stroke. So, I mean, there are other possibilities, but at the moment, current clinical practice type trials, I don't think there's enough evidence to move forward with that.
What would the numbers have had to look like for the trial to conclude that there was evidence of efficacy?
Well, I think based on what we have now, you would need to design a much, much bigger trial because there was only a 5% difference between the placebo and the active group. And actually, it was in favor of the placebo. So the placebo did a little bit better, not statistically significant. So it could just be by chance, but you would need probably thousands of people.
I see. And I imagine there's not a lot of excitement about funding something like that by the people who fund these trials.
Yes. Typically, the funders will look at how good is the evidence to pursue this. And if you were a pharmaceutical company on a pathway to development for a drug, you probably would say, well, it looks safe, but it didn't do what it intended to do. So let's stop the development of this drug for this indication.
I see. So I think one of the challenges with the path of administering etanercept to stroke survivors is that there seems to be a missing step. And the step to me is determining whether or not somebody's a candidate for etanercept. So perhaps if we knew more about the stroke survivor, what was actually happening in their particular brain, and we were able to determine some similarities between the people who have had a positive result, and we developed a method, then that would make it a lot easier to say, well, I'm a stroke survivor, I'd like to have etanercept, and then go through a process of determining whether or not I was a candidate, rather than just guessing whether I'm a candidate or not, and then having to pay money to find out whether in fact I was a candidate.
The trial provides a little bit of answers to that. You want to identify a marker or a subgroup of people in whom the drug will work particularly well. And so you could look at, and we looked at different things like females versus males, if you're younger versus older, if you have very severe disability or less severe disability, if you have diabetes, are you early after your stroke or later, that 1 to 5 versus 6 to 15 category. And we could not identify a group in whom the drug worked particularly well. Now, there's a caveat. When you do a clinical trial, it's really hard to look at subgroups, especially if your trial is relatively small, and the PESTO trial is relatively small. So you have to take this with a grain of salt. But there was nothing really promising group that we could identify. So probably you need other markers if you believe in etanercept as a drug. You would possibly need to look at what are the levels of TNF alpha, the drug, the molecule that actually is targeted. Unfortunately, there's nothing like readily available to do that. Could it be that people with a stroke in a particular location that would work particularly more than in others? But we don't have any real way at the moment to do that.
Okay, so we're assuming that the people who experience an improvement after they've had an etanercept shot, that their markers of TNF alpha were lower or higher, or
well, the theory is that they would have a lot higher TNF alpha. Now, as you know, the premise is etanercept works by reducing this molecule, and we have good evidence that it reduces this molecule in the blood, but we don't have good evidence that it reduces the levels in the brain. That's where you want it to be. And one of the difficulties, and many scientists that work on etanercept and have said, look, it doesn't cross the blood-brain barrier. It doesn't go against the natural defense that we have to protect the brain against substances that could potentially be harmful for the brain or that have a large size. And etanercept, we know, it has a large size, would not cross the blood-brain barrier. So it doesn't reach the brain. And many people look at it with relative skepsis that it actually enters the brain.
Uh-huh. And then with regards to rheumatoid arthritis, it doesn't need to cross the blood-brain barrier. It just somehow gets to the position or the place where inflammation is occurring. TNF-alpha is active, and it can easily mitigate the impact that TNF-alpha is causing. In the brain is protected by the blood-brain barrier, and it cannot cross the blood-brain barrier under normal conditions. And therefore, it can't get to where the TNF-alpha is if there's any TNF-alpha, if inflammation is the issue, and it cannot resolve it one way or another. So for some people, perhaps it can't resolve it. Now, I don't understand about etanercept a lot. I don't understand exactly how the molecule works, etc. But if it was injected into a blood vessel, is that not something that can occur? And if it was, and if it can occur, would that then cross the blood-brain barrier?
They wouldn't cross the blood-brain barrier, no.
Either.
You would have to do what we call a lumbar puncture, or put a little injection into the ventricles, and then hope that it would enter the area that is target where the TNF-alpha is elevated. Those experiments have not been done.
Okay. So a lumbar puncture is probably riskier than...
Well, it's uncomfortable. It's uncomfortable, and we do it to administer drugs if needed. Some people with brain cancer receive it. There are other trials ongoing in certain areas of stroke where it's done.
Then the difficulty is, and my job here is to report back to the community how they should proceed with etanercept going forward. Now, I don't expect you to answer that. However, your study probably gives enough information for people to be able to make an even more informed decision than they did before. Previously, what I think was happening is people, and it still happens every day, and I've interviewed a lot of stroke survivors who've had positive results with etanercept. The challenge is getting interviews with stroke survivors who have had negative results with etanercept. That is something I haven't been able to do. So if somebody happens to be watching and listening to this, and they have had etanercept shots and they didn't get positive results, please reach out so that we can share a balanced story of what's happening out there in the community. Would there be a reason for the community to perhaps begin again to lobby a government or a minister of a government to look at perispinal etanercept and study it in a different way, like administration via a lumbar puncture?
I think we need more, probably go back to the drawing table to see whether, because we just taking a step back, the idea is that there is inflammation after stroke, and we know that there is inflammation after stroke. We just don't know how long it is. We don't have a good marker. Is it present only for weeks or months after stroke, or can it persist for years? The theory is that it persists for years, but if you look at the actual experiments that have been done, it's really hard to study in humans because we don't have good tests. But if you look in animals, it's also hard to do long-term studies in animals. But nobody has really proven that conclusively that there is still, after the stroke causes a scar, that process is still really active. And is TNF-alpha years after a stroke still present? Yes, it's present because we use TNF as a transmitter in the brain or a chemical in the brain, but is it still worth reducing its activity? That's probably, I think, a bigger question that science needs to answer, is to understand that whole inflammation piece and the time after stroke that it persists, in my opinion.
Yeah, because it could still be the fact that the person has had brain damage. A particular part of their brain that is damaged has, for example, taken offline one of their limbs, and there is no way to recover that once it's gone. And there may also be no inflammation there. So somebody in that situation receiving etanercept wouldn't get a result even if it was able to cross the blood-brain barrier, because the damage is done. And that's the challenge with the brain, is once it's damaged, restoring the damaged part is not possible.
Yeah, look, after this experience with the PESTO trial, I think we need to work on other avenues, and I'm not as hopeful with this based on the data that I have seen.
Yeah. Well, my final question then is, are you planning on exploring inflammation and recovery after stroke with any work that you're doing in the future? Is there any more of this type of work being done?
We've just launched a new study, which is not a randomized trial, but it's trying to get at this common symptom that people have after stroke, which is fatigue and cognitive changes. And one of my postdocs, Dr. Emily Ramage, she's a physio by background. We just launched what we call the deep phenotyping study after stroke. And we are looking at young people that have had a stroke up to age 55. And we're taking them into the scanner. We will do a PET scan that's looking at inflammation. We're taking their bloods and looking at markers of inflammation and see how that relates to fatigue after stroke. This is between the first month and the 6th month after stroke. But that will give us a little bit of timeline of inflammation after stroke. It will give us some information about fatigue, which is very common. But I have no plans at the moment to look at etanercept anymore.
Fair enough. I appreciate your time. Thank you so much. All right, well, that brings us back to the end of the episode with Professor Vincent Thijs on the PESTO trial results. My hope is that this conversation gives you more clarity, especially if you've felt caught between personal stories, strong opinions, and a lot of uncertainty. The goal here isn't to tell you what to do, it's to help you ask better questions and make decisions with your eyes open alongside a qualified healthcare professional who knows your situation. If this episode helped you, please do a couple of things. Subscribe on YouTube or follow the podcast on Spotify or Apple. Leave a review if you can. It really helps more stroke survivors find the show. And if you've had an experience you're willing to share respectfully, positive, negative, or mixed, add a comment. Those real-world perspectives help community feel less alone. And if you'd like to support the podcast and keep it going, my book is at recoveryafterstroke.com/book. And you can join the Patreon at patreon.com/recoveryafterstroke. Thanks for being here with me. And remember, you're not alone in this recovery journey.
Importantly, we present many podcasts designed to give you an insight and understanding into the experiences of other individuals. Opinions and treatment protocols discussed during any podcast are the individual's own experience, and we do not necessarily share the same opinion, nor do we recommend any treatment protocol discussed. All content on this website and any linked blog, podcast, or video material controlled this website or content is created and produced for informational purposes only and is largely based on the personal experience of Bill Gassiamis. The content is intended to complement your medical treatment and support healing. It is not intended to be a substitute for professional medical advice and should not be relied on as health advice. The information is general and may not be suitable for your personal injuries, circumstances, or health objectives. Do not use our content as a standalone resource to diagnose, treat, cure, or prevent any disease for therapeutic purposes or as a substitute for the advice of a health professional. Never delay seeking advice or disregard the advice of a medical professional, your doctor, or your rehabilitation program based on our content. If you have any questions or concerns about your health or medical condition, please seek guidance from a doctor or other medical professional. If you are experiencing a health emergency or think you might be, call 000 if in Australia, or your local emergency number immediately for emergency assistance, or go to the nearest hospital emergency department. Medical information changes constantly. While we aim to provide current, quality information in our content, we do not provide any guarantees and assume no legal liability or responsibility for the accuracy quality, currency, or completeness of the content. If you choose to rely on any information within our content, you do so solely at your own risk. We are careful with links we provide. However, third-party links from our website are followed at your own risk, and we are not responsible for any information you find there.